Hi, everyone. My name is Tuomas Tammela, and I'm an associate member at Memorial Sloan Kettering Cancer Center here in New York City. I'd like to start by thanking the organizers for the invitation to present some of our recent work on cell state transitions that drive resistance to lung cancer therapy. These are my disclosures. This support from monopharma is relevant to what I'll talk about, but the others are not. Um, so um, we are in a point in time where we have very good drugs, cancer drugs, chemotherapies, targeted therapies, and immunotherapies that work quite well even in advanced stage disease, where oncologic therapies are the only treatment option for the patient. But as we all know, these therapies invariably fail in almost all cases. And what's really notable about many of these therapies is that, as I said, initially they work very well. We can see very, very robust responses, even complete responses. But invariably, as we've now learned, there is a minimal residual disease that's been left behind that then will eventually reconstitute the disease, cause a relapse. And typically, at that when that happens, there is either a mutational process or some sort of other adaptation, so in some cases, even a very dramatic histologic transformation of lung cancers into different histologies that then leads to resistance. And at this point in time, a second line therapy is warranted. But fundamentally, the disease that now the oncologist and the patient are facing is different from the original disease. And this really is the clinical problem. So what we have been focusing on is the minimal residual disease and finding ways of targeting this stage, this problem really in the clinic. And so the idea here is that if we were able to identify mechanisms that enable these resistant cells to either arise or persist after therapy or even on very aggressive therapy, we were able to identify the particular cancer cell states that are driving this process, then by combination therapies where, for example, cytotoxicity could be directed against these resistant cells, either via cytoablative drugs, such as antibody drug conjugates or CAR T cells, BiTEs. There are many options, as everybody knows these days. We could achieve in combination with standard of care therapies much better responses and hopefully even reach cures. And so we've been studying this extensively in the laboratory, and the lung cancer that we primarily focus on is lung adenocarcinoma, which obviously needs a little introduction with this audience, but it is the most common subtype of non-small cell lung cancer. This is a cancer that in about 80% of cases in the clinic arises in the distal lung, in the alveolar compartment. And really the cell of origin has been now, both in preclinical studies as well as in very elegant genetic studies in human patients, been linked to these alveolar type 2 cells, which are the stem cells, the regenerative cells of alveoli. And as is the case with pretty much every cancer that anyone's ever studied, cancers come from stem cells that have some degree of regenerative capacity. And so the alveolar type 2 cells are the predominant cell of origin for lung adenocarcinoma. And many years ago, we started out with this question: how does this heterogeneity of different cell states emerge in just sort of unperturbed cancer evolution? And needless to say, as everybody knows, tumors are very heterogeneous. We've known that by just looking at using a method that is now almost 150 years old, immunosilent eosin staining. We know that cancer cells within a particular tumor, within a single tumor, can have very different appearances. They can look different from one another. But now, of course, with single cell genomics, we've been able to characterize, we meaning us as a community, the heterogeneity of human clinical tumors as well as tumors in animal models with a very advanced level of granularity. And one of the things that's really notable about these cell states that in this case are transcriptionally defined based on mRNA sequencing, single-cell mRNA sequencing approaches, is that these cell states can, of course, arise through genetic variation within the single tumor. You can have subclones that have particular genetic constellations of mutations, and those can cause heterogeneity in tumors. But the type of heterogeneity that we have been primarily interested in the laboratory is really this cell state heterogeneity that is primarily driven by epigenetic alteration, epigenetic variation, as well as cues from the local microenvironment. And by those criteria and sort of taking those characteristics, those drivers into consideration, it's implicit that cell states are not fixed. The epigenetic state or the microenvironment of the cell can change, and that can then cause a change in cell state. And these changes in cell states are typically referred to as plasticity, so the ability of cancer cells to adopt and acquire different cell states in tumorigenesis, and that in that way promote tumor progression, acquire features that, for example, drive high levels of proliferation or metastasis or invasion, and also drug resistance. These processes are really what we've been studying. And one of the things that many years ago, as I was getting into this field, we realized was that when we profile human tumors in patients with clinically manifest disease, we are always looking at a sort of an end stage of a very long process of cancer evolution. So we typically capture these tumors when they're already very large. They contain billions of cells, and they have many, many different cell states. They're very heterogeneous. However, we know that all cancers come from a single cell. And in the case of lung adenocarcinomas, 80% of them, they come from alveolar type 2 cells, which are the normal regenerative cells. And so this process of how these different cell states evolve remained an open question. And this is a question that's actually quite difficult to address with in the human patients, because it's very difficult to find these very early stage lesions in patients and really study them robustly. And so this is where mouse models, genetically engineered mouse models, can become very helpful. And the model that we used was developed by my postdoctoral mentor, Tyler Jacks, already now almost 25 years ago. And this model is probably one of the most widely used mouse models of any cancer. And it's widely used because of many reasons, but one of them is that it's a very good model, both molecularly and histopathologically, and also in how this disease model responds to various standard of care therapies. It recapitulates the human disease quite well. And so in this model, we use genetically engineered mice. So the mice carry conditional mutations that can be activated by just having the mice inhale virally delivered, in this case, Cre recombinase, and that causes alveolar type 2 cells to undergo a process of transformation. So here we turn on an oncogenic form of KRAS, which is the most common molecular driver, oncogenic driver in lung adenocarcinoma. It's the driver in about 30% of the patients. And now as if we also delete P53, the king of all of the tumor suppressors, as we activate RAS, we allow progression of these adenomas, which are still premalignant lesions, into frank adenocarcinomas. And so this is a very good model that allows us to, in a very controlled way, both spatially and temporally, to investigate cancer evolution. And we did this now many years ago in really what was back then sort of the stone ages of single cell genomics. Already 2013 was when we started this, and this was really a collaboration, wonderful collaboration with Aviva Gev at the Broad Institute and her postdoc Matan Hoffrey, who's a computational biologist. And what we found in this study was that very early on, as we introduced these KRAS mutations, most of the cells still resemble very much the cell of origin. They have these alveolar type 2 cell features molecularly, but some of these cells make this very dramatic leap in phenotypic space. They jump and they change and they lose their alveolar type 2 identity. And instead, they acquire this highly mixed identity. Now, all of a sudden, there are cancer cells already in very early stage tumors that lost the lung identity and instead harbor features of many different cell types all at once, ranging from endothelial cells to chondroblasts to even trophoblast stem cells, kidney epithelium, and so forth. And so these cells are, they were very interesting to us at the time, and they had not been previously seen in any sort of hypothesis-driven study. And what was notable about this cell state was that it continued to be maintained all throughout the progression of lung cancers, all the way to these late-stage adenocarcinomas. And actually, not only was this cell state maintained, its relative frequency increased in the tumors as the tumors progressed. So that really, this sort of novelty of the state and the expansion of the state in the tumors caught our interest. And so we wanted to study the cell state a bit further. And one of the things that we did was next, with Aviv's Natan's help, was to do unsupervised clustering of the cell states. And here we've organized them in the order of their appearance. This cell state, the highly mixed state, shook out in this independent analysis, also as a distinct cell state. And it was the fifth cell state to emerge in cancer evolution. And now if we ask, using a model Aviv's group had developed called Waddington's optimal transport, if we ask now the computer to draw the most efficient path from the alveolar type 2 cell of origin to these late stage cell states that emerge in these very advanced adenocarcinomas, including epithelial mesenchymal transition and then embryonal like states, this cell state, cluster number 5, came out as a cell state with by far the largest number of possible downstream states. And as a result, we started calling this cell state a high plasticity cell state. It really seemed to be the cell state that allows the expansion of heterogeneity as these hyperplastic early stage lesions, which all of us have many of, and they normally don't cause any problems, as they transition in those rare cases when they cause potentially lethal disease, lung cancer, they seem to be going, based on this model at least, through this high plasticity cell state. And we, of course, I told you this, that we found this cell state long ago. And at the time when we found it, it didn't really seem to correlate with any other previously published cellular identity, including cancer stem cells or normal tissue stem cells. It really didn't look anything like that. But we didn't quite believe this was something unique to cancer, simply because over and over again, as we study cancer, we realize that cancer never invents anything new. Rather, it co-opts processes from usually normal, either embryonic development or tissue regeneration and wound healing. And so this is indeed what we realized, because now if we think about physiologic plasticity, it really is driven by adult stem cells. We lose about 60 billion cells every day, and they need to be replaced. At least epithelial cells and hematopoietic cells are replaced by the activity of adult stem cells. And in normal homeostatic conditions, our adult stem cells are actually fairly restricted in their lineage potential. They're capable of giving rise to cells in that particular lineage, but they're not able to cross lineage barriers. However, upon injury, something very special happens to our adult stem cells. They become these super stem cells that actually now are able to cross lineage barriers, and in this way contribute to the repair and regeneration of related lineages that the resting homeostatic adult stem cells wouldn't be able to do. And the lung is actually a great example. Here in homeostasis, the proximal lung, the bronchioles, and the bronchi have their own stem cells. These are the basal cells, basal epithelial cells, that take care of replacement of cells that are lost during normal turnover. And in the alveoli, we have the AT2 cells, which I already talked about. But upon injury, these stem cells from these two compartments they acquire the capacity to now cross lineage barriers and contribute to the repair of the other compartment, which of course increases the robustness of our bodies and increases our regenerative potential. So it's a great feature. It's great that we evolved this. We're not quite lizards that can grow a new tail, but we still maintain a large amount, actually a large degree of regenerative plasticity. And this had been now around the time and a little bit after we had published our cancer work, realized in for example, normal alveolar regeneration, as well as in the intestine and in skin wounds. these types of epithelial epithelial stem cell states that are transient, only present during regeneration, were discovered. And now what we hypothesized was that this very same process that enables this fantastic, wonderful regenerative capacity actually in the context of transformation can become a liability, because now it allows this expansion of heterogeneity, the emergence of these very extreme lineage transitions such as epithelial mesenchymal transition, or these neuroendocrine or squamous transitions that are often also observed, especially under therapeutic pressure in lung adenocarcinomas. And to test this, 3 postdocs, one that's already a PI now in China, Yan, who started it all, Jason Chan, a computational biologist and an oncologist as well, and Chunhao Pan, who's really done a large amount of the experiments, teamed up together to investigate this functionally in our mouse models. And to do this, they chose to take advantage of methods that have been used in developmental biology the world over, one of them being lineage tracing, which is the introduction of a heritable genetic mark, usually a fluorescent protein, into a subpopulation of interest. And then by following the fate of the cells over time, the cells that retain that label, one can infer information about the plasticity of the cell, the differentiation potential of the cell. The other powerful approach is lineage ablation, which really mimics the activity of cytoablative drugs that are directed against particular cell states. So this allows us essentially to subtract a cell state of interest from the tumor and then investigate what happens to the rest of the tumor. And for us to do this, we needed to identify a specific marker gene for this cell state. The gene we landed on is called SLC411. It's a sodium hydroxyl co-transporter. Its function is pretty obscure in various other tissues. But in the case of lung tumors, it was very specifically restricted to this high plasticity cell state. And so we made a mouse model that allowed us to incorporate this tripartite reporter into these SLC411 positive cells. So we can see the plastic cells using red fluorescence. We can do the lineage tracing. We can do the ablation as well. And when we do this, and when we initiate these autochthonous tumors in these mice, we see the highly plastic cells decorating about 15% or so of the cancer cells. And now if we do the lineage tracing experiment early on in cancer evolution, so in the very early hyperplasias, we can see that the trace cells indeed, if we just study the phenotypes the trace cells acquire using single-cell RNA sequencing, the trace cells indeed are responsible for this expansion of heterogeneity that accompanies the transition of hyperplasias to adenomas and adenocarcinomas. And even in late-stage tumors, I told you the late-stage tumors maintain these plastic cells. This cell state continues to give rise to progressively more aggressive states, such as here, an embryonal state that is sort of a very late-stage event. And as a control, we traced another cell state that Aviv's model had predicted to be very fixed, so the opposite of plastic, having little transition capability, that had features of alveolar type 1-like cells. When we trace these cells, we actually see that they behave in a completely opposite way. They actually are very fixed in their phenotypic, in their differentiation capacity. So this really unequivocally, we think, demonstrates that this high plasticity cell state indeed is plastic, as the name implied, but also has functionally a very important role in the expansion heterogeneity in cancer progression. We wanted to test this using now the suicide gene. And so here we're initiating lung tumors. And very soon after we induce the lung tumors in the mice, we kill off these highly plastic cells. And so now we are essentially testing whether we would be able to block the progression of these hyperplastic lesions to advanced cancers. And indeed, this is true. And so we never see, when we kill off these highly plastic cells, we never see these larger lesions evolve in the tumors. We never see these hyperplastic lesions in the lung develop into adenomas. And also we see that there is a complete absence of cells that enter this embryonal like state that's marked by HNF4 alpha. So you don't see those cells at all. So we're essentially locking the proverbial barn door where the tumor, the pre-neoplastic lesion, escapes and becomes a lethal disease. And this really, of course, highlights the importance of this cell state and potentially targeting this cell state in early interception of cancers, which could be easily, relatively easily done in a more focused population such as smokers. Now, another instance where plasticity, of course, is very important for cancer progression is drug resistance. And I talked about this in the very beginning. We have been interested in the origins of the minimal residual disease. And now because our studies had shown that this cell state, this highly plastic state, is really the cell state with the most differentiation capacity during unperturbed tumor progression, we hypothesized that it could potentially be the seed for minimal residual disease, having the ability to quickly transition into something upon therapeutic pressure that is treatment resistant. And so here we're doing an experiment where we have now mice with very advanced lung cancers, much like a human patient would. And here we do the lineage tracing, we label the highly plastic cells very early, or right before we start either cisplatin chemotherapy or KRAS G12D targeted oncoprotein targeted therapy with an experimental drug, MRTX1133, that we got from Mirati Pharmaceuticals through a collaboration. When we do this, and now we have in this particular mouse model the ability to track the bulk of the tumor. And here we're tracking the bulk of the tumor cells that are not in the plastic state. You can see that chemotherapy has a mild effect. It's sort of blunting growth a little bit. The RAS inhibitor shows a very strong response. So you see a inhibition of the bulk of the tumor. But now if we compare this to the cells that arise from the high plasticity state, they actually not only survive the therapy, but actually thrive under therapy. So both under chemotherapy as well as under RAS inhibition, the cells derived from the plastic state actually are capable of, they're much more robust in the face of the therapy, really suggesting that this cell state indeed is the seed for the minimal residual disease. And you can actually see here in this lineage tracing experiment how the therapy, either cisplatin or the RAS inhibition, redirects the differentiation of the plastic cells. This is the baseline. Here you can see that this embryonic liver-like state is really particularly favored, that differentiation state under chemotherapy. And then under RAS inhibition, actually this alveolar type 1-like state is favored. And that was very satisfying to see because we had previously published that this cell state is independent of KRAS signaling. It survives even under KRAS inhibition. And what this data suggests is that a large fraction of the resistant AT1-like cells actually come from the plastic cells under therapeutic pressure. This now, of course, literally begs the killer experiment. What would happen if we were to combine ablation of the plastic cells, which we do first, with these standard of care therapies? And we see these very deep combinatorial responses. We actually can't find in the mice these tumors anymore when we either combine ablation of the plastic cells to chemotherapy or KRAS targeted therapy. So this really now reinforces the idea that this cell state is not only an important driver of cancer progression, so it enables that early expansion of heterogeneity, the maintenance of heterogeneity in advanced tumors, as well as this progressive emergence of even more advanced cancer cell states, such as epithelial mesenchymal transition or these embryonal states. And under therapy, this cell state is the one that begets the minimal residual disease. And if we find therapeutic strategies that would allow us to essentially do the same thing in patients where we kill these plastic cells off and then combine with standard of care, we would expect to see very deep responses. And indeed, this is a very active area in the laboratory currently. We're looking for both antibody drug conjugate-based strategies and as well as molecular targets for small molecule inhibitors to identify drivers of this plastic state and use them in combination with these standard of care therapies. With that, I want to thank everybody. I think I already acknowledged everybody from our lab who contributed to this work. We're blessed with many wonderful internal collaborators as well as external collaborators who are named here. And I also want to acknowledge our funding, which is listed here. And I want to thank again you for your attention and for the opportunity to present here today. Thank you.